All right, so we're going to pick up our second section now in chapter 10 and 11 on mitosis, and we're going to go through and talk now about our four phases of mitosis, which are prophase, metaphase, anaphase, and telophase. And one thing that I always found useful when I was learning this, and I think really teaching it, I've kind of reinforced this point, is that there's really like a certain number of things that happens in each step, that if you can learn the number of things that happen in each step and then quiz yourself, it makes it a lot easier, right? So if you learn that, okay, well, there's prophase has three steps, metaphase has one, anaphase has one, telophase has three, then you kind of quiz yourself and you go back when you're studying and say, okay, well, what is the one main thing that happens during anaphase, or what are the three things that happen during prophase, and so forth. So we're going to start off with prophase, and we're going to look here at, like I said before, three things that happen during prophase that are really most consequential for the cell cycle as a whole. The first two things actually of prophase that occur both generally have the same effect, and that is going to be to make the chromosomes become visible. So before prophase, the chromosomes are actually not visible inside of a typical cell that's in interphase. In order for the chromosomes to become visible, two things have to happen. So one of the things that has to happen is the nucleus has to become it breaks down. We'll say it that way. The nucleus is going to degenerate. It's going to break down. It's going to become really to the point where it's completely transparent, and the chromosomes, in part, are visible for that reason. The other main thing that happens in prophase that makes the chromosomes become visible is the chromosomes become more condensed. They go from being that solenoid form to wrapping around those scaffold proteins, and they take on the most condensed form you're ever going to see during prophase. All right, so three things in prophase. Two of them have to do with making the chromosomes become visible. Number one is taking the nucleus away. Number two is making the chromosomes become more condensed. Now, the third big thing that happens during prophase is that the spindle apparatus, or the mitotic spindle, is now going to form. So something else we have to think about here is we're dealing with a lot of chromosome movement during mitosis, and that's all well and good, except for the fact that chromosomes by themselves can't move. Because, right, what is a chromosome? A chromosome is DNA wrapped around a protein. It has no ability to swim or crawl or fly or do anything else. It has to have help moving around inside the cell. And the way the chromosomes move around inside the cell is by attaching themselves to the spindle apparatus, or just usually what I call the mitotic spindle. So the beginnings of the mitotic spindle are going to form also during prophase. So if we put those three things together, we're going to have, during prophase, the nucleus degenerating, the chromosomes becoming more condensed, and the mitotic spindle forming. Now, that forming of the mitotic spindle is actually a fairly complex process. In animal cells, the mitotic spindle starts off forming what are called asters. An aster just kind of is a generic term that means star. There's a star-shaped structure that forms at each end of the cell where the mitotic spindle begins to form. And those asters form initially as a result of centrioles that are present in the centrosome region of those cells. So now going all the way back to chapter 4, we talked about differences between plant cells and animal cells, and we said that animal cells have these centrioles that help them get ready for cell division. Plant cells do not have centrioles, even though they still get ready for cell division in largely the same way. They do it without the help of centrioles. And actually, we don't really even fully understand how they do it, but that is definitely a distinction between plants and animals, is that animals start off spindle formation using centrioles versus plants, who also produce a mitotic spindle, but do so without the help of those centrioles. All right, so that's our prophase. We are taking the nucleus away. We're going to jump to the next slide here and take a look at a later prophase. All right, so remember that interphase cell where the nucleus was completely dark? Now the nucleus is quite a bit more transparent. We can see these dark squiggly lines inside of here. These are actually chromosomes, which are becoming more condensed. And these kind of blurry pink areas here are the beginnings of the mitotic spindle, which is going to form and eventually attach to the chromosomes to allow them to move around inside the cell. So three things in prophase: nucleus breaks down, chromosomes condense, mitotic spindle forms. And those are really in no particular order. I try to be consistent as I said them here each time I said it, but really you could say them in any order. That's perfectly fine. There's no priority or sequential nature of this. It's all kind of more or less simultaneous. So as long as you know all three things that happen during prophase, you should be good to go. So from there, we're going to move to metaphase. And during metaphase, what we're going to do is to line up the chromosomes on something called the metaphase plate. And the metaphase plate is more or less like the equator of the cell. It's not actually a physical structure. Just like the Earth, we think of as having an equator, but it's not actually physically a thing. If you were to fly south from Ohio and look down over the equator in the middle of the Atlantic Ocean, there is not a dotted line floating in the ocean. There's no actual equator painted on the land anywhere. It's an imaginary structure that we use as a matter of convenience. We imagine it as a matter of convenience. In the metaphase plate, it's really kind of the same thing. The metaphase plate is like the equator of the cell. It's the middle part of the cell onto which the chromosomes are going to become aligned. And so if we look really closely at this bottom picture here, we see that in this pair of sister chromatids, one member's on the right, one member's on the left of the metaphase plate. For these two pairs, same thing. One's on the right, one's on the left. One's on the right, one's on the left. Same thing down here. One member of the chromatid pair is on the right, the other member's on the left. And that's a really important thing to realize. Why they're lining up that way is because eventually this one, this one, this one, and this one are going to move to the right, and this one, this one, this one, and that one are going to move to the left. So eventually what we're going for at the end of mitosis is that each end of this cell, the left side and the right side, are both going to have exactly the same chromosomes. Right? So you're going to have this guy on the right, this guy on the left, and the same for all the rest of them. They're going to line up so that one member of each chromatid pair is on either side of that metaphase plate. So that's really the one thing that happens during metaphase, is the alignment of the chromosomes on the metaphase plate. This is a pretty easy one to see under a microscope. Anytime you look at cells under a microscope and you see chromosomes lined up across the middle of the cell like this, you know it's metaphase. It's fairly easily distinguishable. It doesn't last very long in terms of real life timescales, but it's a very distinguishable one because there's really no other time when you see chromosomes lined up in the middle of the cell aside from during metaphase. Anaphase is also part of the cell cycle where we see just kind of one main step, and that's going to be the separation of the sister chromatids. Right? So the key thing here that takes place in order to separate the sister chromatids is that we have to break down that cohesin protein that was holding the sister chromatids together. Once we do that, once we take away that cohesin protein, now the microtubules that make up the mitotic spindle can begin to pull the sister chromatids apart from each other toward opposite ends of the cell. Just like we see up here, these two chromatids are being pulled apart, these two are being pulled apart, and so on and so forth. All of the sister chromatids are being pulled in opposite directions toward opposite ends of the cell. And again, in real life, this is what it looks like. Really no other time when you see chromosomes kind of like this separating from chromosomes that are up here, aside from during anaphase. So very, very easily distinguishable one there when we see the sister chromatids being pulled apart from each other. We can then move into telophase, and telophase, remember, has 3 steps, but really there's an even easier way to remember telophase, because if you know prophase, you kind of automatically know telophase, because telophase is the exact opposite of everything that happened during prophase. So in prophase, one of the things that we did was we built a mitotic spindle. In telophase, we are going to take apart the mitotic spindle. We're going to undo it, take it apart, break it down, and use those microtubule proteins to do other stuff inside the cell. In prophase, we took away the nucleus. In telophase, now we're going to return a nucleus back around both sets of chromosomes now that are on each end of the cell. So now we're going to return the nuclei to the cell. And so by the time telophase is done, you'd actually have a single cell with 2 nuclei inside of it, and each nucleus would have a full set of chromosomes. And the third thing that happened is in prophase, we condensed the chromosomes. In telophase, we are going to decondense the chromosomes. So everything that we did back in prophase, we are now undoing here in telophase. All right, so we'll take a look at that. Something like this, where we've got our whitefish blastula cells. We now have a nucleus reforming here, a nucleus reforming there. Mitotic spindle is being broken down. Chromosomes are now less condensed. You can't really see that too well, but they are being decondensed. And the other thing that's happening here is the beginnings of cytokinesis. So even though technically we still have in this picture a single cell, barely, it is very close to being split into 2 separate daughter cells. So eventually this top part will become one cell, and the bottom part will become a separate cell also. So there's actually a lot of overlap between the end of telophase and the beginnings of cytokinesis when the one cell begins to split physically into 2 cells. So that's cytokinesis then. We're going to split the cell in half. So if you remember, mitosis was nuclear division. Cytokinesis is cytoplasmic division, where we're going to split that one cell into 2 cells. In animal cells, this involves the formation of something called a cleavage furrow. Cleavage furrow is sort of an indentation in the inside of the plasma membrane that eventually constricts and pinches that one cell into 2 cells. Plants, on the other hand, and really fungi are the same way, most protists as well, form what's called a cell plate. So a cell plate is kind of equivalent to what we saw back when we did binary fission with the actual splitting of the one cell into 2 using a septum. Plant cells really kind of do the same thing, where they create vesicles that all contain cellulose, and they stack those vesicles up in the middle of the cell until eventually they have a full-fledged cell wall, dividing that one cell into two. And for them, that's what's going to allow them to complete cytokinesis. Fungi and protists are a lot more variable. A lot of times, especially fungi, they can actually go through a bunch of rounds of mitosis without doing cytokinesis. So what you can actually end up with there is a fungal cell that has maybe 2 or 4 or 8 or 16 or 32 or 64 nuclei. Because it's gone through a bunch of rounds of mitosis, it's divided the nucleus over and over and over without actually doing cytokinesis. So you get multiple cells, normally that would form in an animal, actually form as a single cell in a fungus. So one cell with lots of individual nuclei. This is an illustration of what a cell plate would look like. We have the one cell, chromosomes at each end, cell plates forming across the middle of it, until eventually we see a complete separation of these two cells by this cell wall, which is now formed in between them. So now two cells, identical nuclei, but separate from each other. These two pictures illustrate a cleavage furrow in animal cells. So the cleavage furrow is wrapped right around here in that pair of cells and right around here in this eventual pair of cells. Really what's doing this are the actin filaments that are inside the cytoskeleton. All right, so again, if you go back to chapter 4, look back at the cytoskeleton, one of the functions of the actin filaments, the microfilaments, is to help in cytokinesis. And so what's happening is that inside this plasma membrane, there are actin filaments that are attached to the membrane, and they are going to begin to shorten. And they're going to get shorter and shorter and shorter, and that's going to squeeze this membrane tighter and tighter and tighter until eventually this one cell, which is constricted in the middle, is actually going to split into two separate cells that each has its own nucleus now. All right, so that is the cell cycle. As we're going to look at it, as far as the depth that we're going to look at it when it comes to mitosis and cytokinesis, we have a growth phase 1, we have DNA replication, we have a second growth phase, and then we have our prophase, metaphase, anaphase, and then telophase and cytokinesis, which obviously overlap quite a bit. For the rest of this chapter, we're going to talk about the control of the cell cycle and also what happens when the control over the cell cycle fails and we end up with a situation called cancer. All right, so like I said before in the previous section, there is a lot of difference in the amount of time it takes cells to go through G1 depending on what kinds of cells we're dealing with. So skin cells and intestinal cells and stomach cells, those guys all divide pretty quickly because they have to be replaced fairly often. On the other hand, brain cells, spinal cord cells, cardiac muscle cells in the heart, skeletal muscle cells, all of those cells really divide rarely, if ever. It's really actually more accurate to say they never divide than they divide even infrequently. There are some that do divide a little bit, but it's a very, very low rate of cell division. So clearly then, because these cells all originally started off from the original zygote cell, right? They're all genetically the same. There has to be something about them that's controlling their ability to go through the cell cycle at different rates. And so one of the things that controls how the cell cycle works is called a checkpoint. And so the checkpoints are places in the cell cycle where the cells can kind of be put on hold as far as the cell cycle. And if it contains any damage, if it's flawed, if it has a mutation, if it's in any way abnormal, the cell can either be repaired or destroyed instead of having that one faulty cell divide into two separate cells. Now, those are checkpoints. There are also what I kind of like to think of as points of no return in the cell cycle, where once a cell has gone past this point, it's not able to go back again. All right, so one place in the cell cycle that is a point of no return is replication of DNA. Once a cell has gone through S phase, once it has those two now completely identical genomes or sets of DNA, it kind of then has to go on to the next step. It kind of has to go on to G2 and eventually to M phase, because that one cell cannot persist and function normally having all of that extra DNA inside of it. So once a cell goes through S phase, it is pretty much irreversibly committed to go ahead and go on to the next step. The other place where we have a point of no return is the separation of the sister chromatids. And if you remember back to our discussion of mitosis a few minutes ago, that is during anaphase. So once the sister chromatids have separated, once that cohesin protein is broken down, then those two sister chromatids are committed to go ahead and move to opposite ends of the cell, and from there telophase will take place accordingly. All right, so those are points of no return. The checkpoints are places where, again, like I said, the cells can be screened to make sure that they're still normal, they're still functional, they're still behaving the way that they should, and if there are mistakes or if there are errors, the cell can either be fixed or destroyed, rather than propagating those errors and allowing those cells to go ahead and divide more and more and more. So one of the ways that all this is controlled, and there are many, many levels of control over the cell cycle, is through the use of a group of enzymes called cyclin-dependent kinases, or CDKs. So CDKs are enzymes that have the ability to either phosphorylate or dephosphorylate proteins that are involved with, in this case, the cell cycle. They can do it in other parts of cellular function as well, but here we're talking about cell division and cell cycle. These enzymes have the ability to phosphorylate proteins or dephosphorylate proteins in the cell cycle, thereby either deactivating them or activating them depending on how that cell normally functions, either with a phosphate group or not. So these are kind of like on-off switches that allow the cell to control when different parts of the cell cycle actually take place. So we'll look at some examples of that here in a second. Here are some things we've already talked about in the cell cycle, right? So the synthesis and activation of proteins required for DNA synthesis. So what are we talking about there? Well, DNA polymerase, DNA primase, DNA gyrase, and helicase, and ligase. All of those enzymes can be activated or deactivated by cyclin-dependent kinases, thereby either favoring or not the cell cycle, moving on to the next step during DNA replication. The condensing of the chromosomes, the breakdown of the nuclear envelope, forming the mitotic spindle, and attaching the chromosomes to the spindle, all of these things happen during prophase. So CDKs are also a way to control all of the various things that happen during prophase, either allowing them to continue or putting them on hold and allowing a cell to be screened to look to see if it has damage and whether or not it should be fixed. And then also separating the sister chromatids during anaphase to allow them to move to opposite ends of the cell. That also can be controlled using CDKs because the enzyme that breaks down the cohesin protein is able to be phosphorylated and dephosphorylated by a CDK. So that is another way to control a part of the cell cycle specifically and allow it to either move on to the next step or to be put on hold. All right, so we're going to skip that. And we're going to skip this as well. And we're going to move on to our checkpoints. All right, so our specific checkpoints, number one, the G1 to S checkpoint. So between growth phase 1 and DNA replication, there is a checkpoint where cells can decide whether or not they're going to divide at any given moment in time. This is also a point in the cell cycle that's very much subject to control from, like it says, external signals. So materials coming in from other cells in the body, other tissues. The energetic state of the cell is also a big driver of this. So if a cell is in good shape energetically, it is going to be more likely to try to go ahead and divide than if it's struggling or if it's sick or if it doesn't have enough energy. Then it's probably not going to get through mitosis anyway, so it would not be a good idea to make that cell try. So this is a place where there's a lot of opportunity for control and contribution from other areas outside the cell as far as whether or not cell division should take place. We also have the G2 to M checkpoint. So at the end of growth phase 2, but right before mitosis, there is also a point where a cell can make a decision whether or not it's going to divide, primarily as a result of how well DNA replication went back during S phase. Because remember, what comes before G2 is S phase, DNA synthesis. And so before we let the cell go ahead and divide to form two cells, we want to make sure that it was able to successfully get through DNA replication without any mistakes being made. So the DNA will be screened, it'll be proofread. If there are mistakes, hopefully the mistakes can be fixed. If they can't be fixed, then the cell will be destroyed. Because you're better off to destroy one faulty cell than to let that cell divide and then have two faulty cells. Our third checkpoint is a late metaphase checkpoint, and this is a point where cells are screened to make sure that everything lined up correctly on the metaphase plate, to make sure that we have one member of each pair of sister chromatids on either side of the metaphase plate, so that eventually both daughter cells that are produced will be genetically exactly the same thing. That's also a checkpoint. If we have chromosomes lining up incorrectly, if we have, for example, two chromatids on the same side of the metaphase plate, then again, that cell could be fixed, chromosomes corrected, or if that doesn't work, then the cell can be destroyed as well. All right, so these are sort of our checkpoints sort of overlaid on the overall cell cycle from G1 to S. Before we copy the DNA, we really want to make sure that there aren't any mistakes in it. There aren't any mutations or anything like that. And if there aren't, then we can go ahead and copy the DNA. We can move on to G2. But before we finish G2, we want to make sure that DNA replication happens successfully back here, and there weren't any mistakes made when DNA was being copied. And then if we go into mitosis, we also have that late metaphase checkpoint to make sure that everything is lined up correctly on the metaphase plate before the chromosomes separate and move to opposite ends of the cell. Now, if there is a mistake in any of this, if there is a mistake in the cell cycle, if there's damage to a cell, it is possible that that one cell could become a bit of a problem and become cancerous. So this last little section of Chapter 10, and we're actually almost done with Chapter 10, is to look at, just in a nutshell, in a very, very short maybe 5 or 10 minute description. What exactly is cancer? Right? So cancer is defined essentially as out of control cell growth. There is a lack of control over the cell cycle, and so cells begin to grow faster and faster and faster, and they divide faster and faster and faster, and they lose their functionality. They can no longer do the jobs they're supposed to do because they're so focused on just one thing, and that is dividing. And so it turns out there are really two kinds of things that typically precede cancer as an effect in the body. Number one is a failure of what are called tumor suppressor genes. So tumor suppressor genes are like the brakes in your car, right? So if you get to go too fast, your brakes can slow you down. The same thing with tumor suppressor genes. If cells start to grow a little bit too fast, if they start to divide too quickly, if they start to fail to carry out their normal functions and just try to divide over and over and over again, tumor suppressor genes have the ability to make that stop or prevent it in the first place, is another way to think about this. So if you have damaged tumor suppressor genes, then it's like you've lost the brakes in your car and you can no longer slow down, in this case, cell growth. The other thing that usually precedes cancer is the activation of what are called proto-oncogenes. So the term proto-oncogene means literally it's like an early form of a cancer gene. So the first thing that I always like to clarify here is that there are no such thing as true cancer genes. And you'll hear that reported in the media all the time about how researchers have discovered a cancer gene for blah, blah, blah. There is no such thing as a cancer gene. Right? If you think about this even logically, very simply here, if there was a gene whose only job in life was to give a person cancer and kill them, what would happen to that gene in the population over time? Gene would go away, right? No more gene. So therefore we wouldn't see it anymore. But we do. So proto-oncogenes actually are genes that have other functions in our bodies, but if they're altered, if they're damaged or mutated, they begin to take on other functions, specifically in the realm of making our cells divide faster and faster and faster. So proto-oncogenes are kind of like the gas pedal in your car. You push the gas pedal, you go faster. You activate a proto-oncogene, cell division occurs faster. Now, either one of those two things by themselves, either a failure of tumor suppressor genes to function, or the activation of proto-oncogenes, and we'll kind of explain those more here in a second. Either one of those two things by themselves is bad enough, but if they both occur together, that's like a worst case scenario. So if your gas pedal sticks to your floor, your accelerator is stuck to the floor, and your brakes no longer work, then chances are you're going to have a pretty bad day. If it's one or the other, like if your brakes fail, but your gas pedal's fine, you could still kind of slowly slow down and maybe be okay. If your gas pedal sticks, but your brakes still work, you can still slow down. It's not going to be as easy, but you can still slow down that way. But if your brakes fail and your gas pedal sticks to the floor, now you're going to have a kind of a bad outcome to that particular circumstance. And that's exactly what happens with cancer, is that both of those two situations occur simultaneously. Tumor suppressor genes are lost and proto-oncogenes are activated. So one really good example of a tumor suppressor gene is the gene called P53. Right? Some of these genes don't have some real creative names. This one's just alphanumeric P53. But this particular gene is really important in playing a role as a checkpoint protein down here in the G1 to S checkpoint. So prior to a cell being allowed to copy its DNA, the proteins produced by P53 are going to screen the DNA to make sure there aren't any mistakes there. And if there are mistakes, if a cell's damaged, those P53 proteins can signal for repair enzymes to come in, hopefully fix the DNA, and then allow the cell to go ahead and divide like it was planning to anyway. On the other hand, if P53 proteins find out that the damage is irreparable, if it's too severe, then that P53 protein will tell the cell to commit apoptosis, which we've talked about a little bit before, but apoptosis is programmed cell death. This is cell death on purpose. This is cell suicide, essentially. So if the cell can't be fixed, the P53 proteins tell the cell to kill itself, which seems kind of harsh, but you're much better off to lose one cell that way than to lose the entire organism because you have an outburst of cells dividing out of control. So P53 is one example of a tumor suppressor gene that is very commonly lost in the development of cancers. Now, the thing about P53 that's kind of nice is you really only need one functional copy to get protection from it. So even if you have a good functional working copy that you inherit from one parent, but the other parent gives you a broken copy of the gene, you still have protection from P53. You only need one functional copy of the gene to be protected. Now, if something happens to that one functional copy in some random cell throughout your body, then you might potentially be at a higher risk for cancer. It doesn't guarantee cancer, right? So just like having a brake failure in your car doesn't guarantee you're going to be in an accident, but it does increase the risk, right? So having two broken P53 genes in a cell doesn't guarantee a cancerous outcome, but it greatly increases the chances. you're much better off to inherit two working copies, from one from each parent, so that if something happens to one of those working copies in a cell at some point in your body, you still have the other working copy to protect your cells from this particular circumstance. So this is the idea with p53. We've got some DNA damage here on this particular double helix. We've got the DNA repair enzymes calling in for help, and the damage is fixed. The cells are then allowed to divide, and the cell cycle goes on as it would have otherwise. If the damage is not repairable, then the cell is told to kill itself, and it goes through this programmed cell death where it's removed from the consideration of cell division. So it doesn't propagate itself, and you don't end up having millions and millions of faulty cells in your body somewhere. On the other hand, if you don't have p53, you don't have the protein, you at some point develop an abnormal cell, and now there's nothing there to tell that abnormal cell to fix itself or kill itself. So eventually that faulty cell begins to divide over and over and over and over again, and before you know it, you've got a tumor growing somewhere in your body, a whole mass of out-of-control cell growth that eventually becomes lethal, potentially. Which is, I guess, something else that's important thing to think about as well, is why is cancer, how does it hurt you? Right? So the way the cancer works is if you have a tumor growing somewhere in your body, that mass of cells that's growing out of control, they're very, very hungry. They need energy to divide. And not only that, but they're not really doing much else aside from dividing. So that patch of cells that you have growing out of control is going to start to siphon off more than its fair share of resources. It's going to use more oxygen, it's going to use more glucose, it's going to use more nutrients and produce more waste than the other cells around it. So the other cells around it that are normal, the normal tissue cells or organ cells you have in your body, they stop working because they're not getting the resources they're used to, because all those resources are being funneled into the cancer growth. And eventually the organ damage sustained from that is a result of them being starved for those nutrients that are instead being devoted to the cancerous cells. All right, so another tumor suppressor gene is one called the retinoblastoma susceptibility gene, or the RB gene for short. This is a much more specific one than p53. This particular gene, it works kind of the same way, but it's very specific against a certain type of cancer called retinoblastoma. So this is a type of cancer that forms on the retina of the eye. It's almost always inherited. It almost never occurs aside from, genetically inherited from parents. And usually when it happens pretty early on in life, maybe 2, 3, 4 years after birth. It's going to start showing up in the children. So in this case, kind of the same idea. If you have two working copies, great. You've got a backup copy that if something were to happen to your other good copy, then you still have a spare one that will help prevent retinoblastoma. But if you inherit only one good copy and something happens to that one good copy, then all of a sudden you're much more susceptible to retinoblastoma. It doesn't guarantee that you're going to develop cancer, but if you have cells that begin to act erratically and you don't have the RB gene at all, now you can't fix those faulty cells, and now those cells are going to grow out of control. Right? So it's not that losing this tumor suppressor gene causes you to have cancer. It's like you're losing the protection that prevents you from getting cancer. So once you lose that protection, any other mistakes that are made could lead to a cancerous type situation. Now, the other situation that I talked about, the gas pedal example, was the proto-oncogene. And I made the statement before that there are no such things as cancer genes, and that is true. But there are other genes that, even though they have normal functions most of the time, if they are changed in some way, they can then become cancer genes. So that's why we call them proto-oncogenes. They're like pre-cancer genes. They do have normal functions in our bodies. We need to have them all the time. But if they change for some reason, they can result in cells growing again out of control. All right? So a really good example of that is in cells in your body that produce what are called growth factors. Growth factors are chemicals that are produced by some cells that tell other cells when they should divide. So you can imagine if you have a mutation in a growth factor producing cell that causes it to make more growth factor than it should, right? So more growth factors produced, all the other cells around it begin to grow faster and faster and faster, divide more and more and more, and before you know it, you have a whole mass of cells whose only job in life, whose only goal in life is to divide over and over and over again. Now, with this situation, it's a little bit scarier because you only need one faulty copy of the gene in order to have the proto-oncogene effect. So while most of us would inherit two functional working copies of a gene to make growth factors in a certain cell, if there's a mutation that affects one of those working copies, then that cell will very likely overproduce growth factor and therefore induce cancer in the cells around it. So this is a slightly more serious scenario. But again, if you have the working tumor suppressor genes, they can also work against this process, and everything kind of evens itself out. as long as you have the tumor suppressor genes, even though you may have activated proto-oncogenes, it might not be a huge effect because the tumor suppressor genes can control the rate of division of the cells that are overproducing these growth factors. All right, so with that, we're going to wrap up this section. That's going to lead us into chapter 11, which is on meiosis and the sexual life cycle. So we'll stop this section here, and I'll pick up with another recording here shortly.